Hello. Hi, I'm Marcelo. This is Gabriel.
Hello, everyone.
Today we're going to talk about this two programs here that we have been working since 2016, I think. And then today we're going to tell a story about how to build maps and map QTLs in interconnected F1 families in hexaploid sweet potato. So each one of these full seed families are hexaploids, okay? And after that, Simon and Guy will talk about some applications of these QTL mapping procedures here. And so I think in the next four talks or three talks, we will have like an overview of sweet potato work in our group at NC State. So, and just let me say, Guy, if you want to say something, interrupt me, because Guy is a huge part of this implementation as well, okay? Okay, so, yeah, I need to stay here. So I know why people don't use this, because, yeah, okay. Yeah, I know. Yeah, I'm not going to use this. Good. Yeah, no, it's fine, it's fine. So let me present you our three biparental populations here, and I'm blind. Let me show you here. We have three biparental populations, and we have three parents: Beauregard, Tanzania, and New Kawogo. We have a cross between Beauregard and Tanzania, Tanzania, Beauregard, and Beauregard and New Kawogo. And we developed these mapping populations in the context of these two projects. They are funded by Bill and Melinda Gates Foundation, called Street Games and GT4SP. The two PIs were Hugo Campos from the International Center for Potato and Craig Yancho from North Carolina State. And so in these three populations, what we did, we genotyped these three populations with GBS, and we used this application called MAPpoly, developed by Bodhi Olokulu from Tennessee. And in this three populations, if you see here in this picture, for BT we have almost 300 individuals. For TB we have 200 individuals, and for NKB we have 255 individuals. And these are the three parents here. So Beauregard is a sweet potato from the US. It's a orange flesh sweet potato with a lot of beta carotene. And Tanzania and New Kawogo, they are from Africa, and they have less beta carotene, almost nothing of beta carotene, and they have high contents of dry matter starch. And I always like to show this picture here. So this is a cross between these two potatoes. See the huge variability you have in an F1. So this is for someone that works with maize or soybeans. This is a crazy amount of variation in an F1, right? And it's mostly because it's a highly heterozygous hexaploid, right? So these are our three populations. And the map for the BT is in this publication here. Okay, so the first thing I want to tell you is, in this context here, we are building maps for each one of the populations. And then after that, we are going to combine those maps, not just putting those maps together in terms of order of markers, but we are going to use the joint map to compute the haplotypes for the hexaploid progeny. And in this context, it's very important that we discover the allelic variants and do the dosage calling for the whole population, because we need SNPs in common across all these populations to merge maps, right? And in this case, we consider all the 728 individuals combined, and we use the tassel4poly, which is something that Guy implemented, which is a modification of tassel for polyploids. And we use the diploid sweet potato genome from Ipomea trifida. Yeah, that's the name. And After we discovered these variants, we performed the dosage calling using supermassa, and we use the structure for each one of the full SIB families for doing that genotype calling. We got the probabilistic dosage calling for all those markers. We have some filters in this part of the analysis. We filtered for minimum read depth of 20. Don't forget, this is an hexaploid, so we need to be very stringent. Our bar needs to be set a little bit higher than we are used to. And markers that don't reach at least 80% of or 0.8 of probability, we consider as missing data. And here we have the three populations, and we have the number of markers per class of marker. Simplex in parent 1, simplex in parent 2, double simplex, and multiplex markers. So, and this is the total number for each one of the populations. And as you can see here, you have a connection between these markers. You have 60K markers that are shared among these three populations. And you have some markers shared between pairs of populations and some unique markers that are not shared. They are just present in that population. This is the raw data set. No filtering other than these two filtering things that we showed here. However, we have to do some more filtering with the mapping structure with the F1 structure, considering the mapping in F1 structure. We usually have two types of filtering. The first one is for missing data. So in this case, it varies depending on the population, but from 20 and 25, if we saw 20 and 25% of the markers with missing data, markers with missing data, we eliminate those markers. And if you observe individuals with more than 18 and from 18 to 22%, we remove those individuals as well. And this is the first part of the filtering procedure using the MAPpoly, or the structure of the population. And also, as they showed in the previous presentation, we also use the segregation distortion filtering, and we use the Bonferroni correction for the individual p-value. And here we have some interesting graphics about what markers we are using in our map. So in the x-axis, we have the read depth for the SNPs. In the y-axis, we have the density. So as you can see, these two colors, the blue were the included ones. So we included the markers in the blue graphic, and we removed markers, we filtered out markers in this red graphic. And the most interesting thing here, as you can see, the markers that we excluded were the ones with less reads, right? With less depth. And if you select just the blue markers here, this is also very interesting because as you can see, the simplex markers and the double simplex markers, they are usually concentrated in the lower end of this graphic, in the lower read depth end here. And for a good multiplex marker, again, remember, this is an hexaploid, you have to have around 400 in terms of read depth, or in this case, 200, and here is also between 200 and 400. And of course, you can have markers, simplex markers, with more read depth, but having less read depth, you still can find simplex markers. And as I said, here what we did, we call these markers, but we have the separate populations. So you can build the map for each one of those populations, right? And in this case here, I'm showing you the recombination frequency matrix for each one of the 15 chromosomes, 15 linkage groups of sweet potato, and in our three populations. Just let me move this. Okay, so this is BT, TB, and NKB, our three populations. In this case, we used the order of the published map, the BT map, which is basically using this first order here, and also the Trifida genome. We have a mixture of these two sources of information to order the markers, and we computed the recombination fraction. And in this case, one thing I want to show you, the difference. So Chris talked a lot about the quality of the recombination frequency matrix. And I think this is very important because you can see here, BT is a population where our read depth is much higher. The coverage when we are doing the GBS was much higher because these are our gold standard populations, our flagship population. So much so that we are using this population to phase the hexaploid genome for sweet potato. And if you want to see there will be a presentation in PEG. And so in here we have our second population. This is chromosome number 1, okay? Just a zoom in the first column of that previous figure. And here, as you can see, you still have a very good shape of the monotonicity of the matrix, but still it's not as good as the first one. And here for the last one, NKB, you can see this shape as well, but it's not any closer than this first population here. But anyway, you can build your map, right, in each one of these populations. The difference here will be that you have much more markers in BT because we had to be more stringent in TB and NKB, but you still can manage to build your map, right? And once you have these maps, what you're going to do is to, of course, you need to phase each one of those maps. In this case, we used MAPpoly, as we showed in previous conferences or opportunities. And having that map, those 3 maps phased, what you can do is to combine these maps. Okay, so what I'm going to show you here is how to combine these maps. You need 2 things, as I was saying. The first one is order, and the second one is phase. You need the phase and the order of the markers in this. The way we did was, as I already said, the order was using the published map. Now, and we are working in another way to do this, which is using the MDS, but to use the MDS algorithm, you need the recombination fraction, a joint recombination fraction for the whole population. We didn't implement that yet, but the idea is quite straightforward how to do that. And another thing that I tried, it really worked, was to use the LP merge, which is a software from Jeff. And it worked very well, meaning that, and why it's important to have the order, because you need to combine your maps. And once you have the order, you need to phase. And as I said, we phased using MAPpoly for each one of the maps. And one of the things we need to do is to assess the correspondence between homologs in one map. So you have Beauregard in one map and Beauregard in the other map. You need to combine or to assess the correspondence between these haplotypes that are in one population and the other population. And at this stage, we also filtered some inconsistent markers. They're very little, less than 1%. So I didn't bother to show you here, but it's very little. And another option to do this would be using polyorigin. Of course, if you have your maps, you just can use polyorigin. But as far as I know, it's implemented for diploids and tetraploids. I talked to Chelsea, and I know he has hexaploid research codes. So this could be another way to do this. But we did with our approach. And some of you might have this question in your head: why are you combining maps? Why it's important to combine maps? And there's a lot of implications combining maps. And one of the things that I really like, this is more about the mapping part, not about the QTL part. Of course, if you want to have QTLs done in multiple populations, you need to combine maps. So in this case, if we have 3 separate maps, the propagation of information would be like this. You see, we have our first population BT, we have more markers. Each one of these dots here represent markers or information in a more general sense. And we have TB in an intermediate situation with less markers, and we have NKB with less markers. If you want to join all these maps, you want to propagate your information in something like this. And we do this using something we call hidden Markov model. Of course, within each population, we have the propagation using the hidden Markov model. But in this case, what we can have is the propagation of information across populations, which is, instead of just using one population, you can fill this transition space with a sort of imputation. You can, if you don't have a marker in one population, but you have in the other one, you can use the hidden Markov module to populate the whole chromosome space. And that's what we did. And in this context. So, and the idea about how to do this is in this publication. It's not for multiple populations, but if you, it's just like it's not trivial, but it's kind of it's an extension of the idea that we published here in 2019. And of course, if you have this, you can have your consensus map. And the most important part is not just you having the consensus map. We are going to estimate the haplotypes or the probability of the haplotypes in the F1 population using all individuals, the 700 individuals together. Instead of just using 200 individuals for NKB, we have meiosis from other populations helping to build those haplotypes. And basically, the code for this is very straightforward, but you need to have the map first. And this is something that we are still working on, how to do this without having a map, or how to do this in a just running in one in one batch. So, but it's kind of difficult, but we're getting there. But today I'm going to present you what we have so far. And in this case, if you use these functions that are implemented here, they're in a branch in my MAPpoly GitHub. It's not in the main branch. There's a branch there where you can pull these functions. And they're still experimental. So if you're going to use these functions, please use with caution, okay? And if you want to contact me to see how to do this stuff, of course, you're more than welcome. And in this case here, I have an example of the first 5 centimorgans in our 3 populations, in the chromosome number 1 in our 3 populations. So we have BT here, TB here, and NKB here. Okay, the first, as you can see, there's more markers on BT, less markers in NKB, in TB, and less markers in NKB. And what you want to know, what is the correspondence between homologs in this first parent here, Beauregard, this first parent here, also Beauregard, and Beauregard here. And what you can do is just use a classical UPG-MEA clusterization with if you just binarize this homology group here, you can reach this kind of very clear cut homolog correspondence. So here you have D1 in the first one correspond to E1, E2 in the second, and D3 in the third one, and so on. This is for this other chromosomes, A1, A2, and A3. Here we are the same homolog. The homologs are the same in the 3 populations, and so on. And then you having this correspondence, of course, you can have, this is for the other, the Tanzania, and Beauregard, you just have Beauregard in 1 population, so we don't need to worry about that. And basically to do this, you need to have your MAPpoly maps constructed, the list of, this is for each chromosome, okay? So this chromosome 5 actually is not chromosome 1. So you have the chromosome 5, you make a list with the chromosome 5s from your several maps, and you can have as many maps as you want here. You know, you can get, I tested with like 20 maps with simulations, but I just don't want to show simulations, but you can do this. And the computational time is linear in this case, which is very important. You need to have a matrix for the parents. In this case, we have parent 1 and parent 2, and the populations of each one of these parents are in. In this case, we change. We have population 2 is TB, but in this case, we have BT. We just swapped the parents. You need also a list for the data sets for each one of these maps, which is straight from MAPpoly. And then you just run this function to prepare to merge maps. It will do all this stuff here of seeing which homologs are the homologs that has the correspondence between or among populations. And then having this procedure, this object here, you just run this like HMM map reconstruction, and you will reconstruct a map for the whole population, right? And this is our final map. I'm just gonna show you. So we have our 15 chromosomes. We have, oh yeah, just let me see. Yeah, so this is the whole map, but I'm going to zoom in chromosome 1. So here you have our 6 haplotypes for each one of the parents in this joint population. Here in red, we have markers that are common among the 3 populations, markers that are common just between BT and TB, same here for TB and NKB, and same here for TB and NKB. And here are markers that are unique for each one of those populations. So as you can see here, you can combine all those markers in just one map, right? These are some statistics about this map. So we have more than 28,000 markers. This is the total length of the map, the maximum gap for each one of the chromosomes here. So it seems that the map is okay. And just to compare, in our isolated populations, we had 22K, 12K, and 8K, 8.7K in terms of markers. And when you combine, you can put all those markers together. But this is the most important part of this, I think, because you can assess each one of the individuals in this combined population using their haplotype probabilities. Just let me explain here. We have 6 haplotypes of Beauregard, 6 of Tanzania, and 6 of New Kawogo, red, blue, and green, right? And here is the probability of inheritance of each one of these haplotypes for a specific position in the map. Here we have the map, right? So for this first position here, imagine a line here. We have a high probability for this dark red, higher probability, almost 1 for this red here, and also for this red. In this case here, we have a change of probabilities, meaning that we had a crossing over between these 2 points here, and you can use the same reasoning for the rest of the chromosome. So we can assemble the whole population structure in terms of haplotypes. Of course, this is also implemented in polyorigin, as I said. In this case, we did this. We didn't use polyorigin because we have an hexaploid thing here, and I'm also implementing our functions to mixed ploid levels in multi-allelic scenarios. But the final result of the map is the map itself, but this is the final result. I just can't, yeah, I can. So here you have your marker. This is a huge data frame, big data frame. Here you have your marker. Here you have your homolog, BR1, Beauregard 1 to Beauregard 6, our 6 homologs. In this case, you have the individual, the probability of that specific allele, and the position in the map. And this is what we use to perform the QTL analysis, which is what Gabriel is going to show us how to do it. Okay, so Gabriel, oh, Guy, you have a photo there.
Yeah, thanks, Marcelo. Hello, everyone. Can you hear me well? Okay, so as Marcelo mentioned, I just show you the rationale behind the strategy that we have been using to map for QTL in multi-parental populations, and some results we got in these three populations, BT, TB, and NKB. So before we jump into the results, I just want to mention that our strategy was based on the random effect multiple interval mapping that was proposed by Professor Guilherme Pereira. We are very lucky to have him here participating in the workshop today. And if you want to get more details about modeling and the procedure, just talk to him or read his paper. And you can also feel free to send us a message or meet sometime so we can talk about that. So everything that we will talk about and the functions that we use to perform this analysis are implemented in the QTLpoly. QTLpoly is an R package which is available on CRAN and also on GitHub on the development version. So the package has implemented both the fixed and the random effect approaches to map for QTL. And it also supports for phenotype weights. So if you have missing plots or even unbalanced trial designs, you can make use of this phenotype weights. We have a website of the package, so you can find materials, tutorials, and also an example data set where you can run and test the functions, all the functions, and see how the package works. And as Marcelo mentioned, we have been working on development of such tools, MAPpoly and QTLpoly. So we have new features coming up. Some of them are supporting for single-step analysis, support for multi-trait, multi-environment, and also multi-parental data sets that support to analyze and perform QTL mapping in those types of scenarios. I won't run through the codes today because the functions are still in the development function, so they are not as user-friendly as we would like them to be. So we will soon be releasing a version of both MAPpoly and QTLpoly with very user-friendly functions for you to use. So just recapping the multiple QTL mapping strategies. So we start with a model with no QTL on it, where all the phenotypic variation belongs to the environmental variation or the residual variation. And we perform a forward search. We start to search for QTL at every genomic position alongside the genome. And for every position, we take the probabilities that Marcelo just showed us how to calculate and how to obtain. We construct a genomic relationship matrix for this position between all the individuals. And then we estimate the variance component associated with that position. So we keep doing and searching alongside the genome. And we have, of course, we have a threshold for that. And this threshold, we usually use the score statistic resampling method. At this time, we use a more relaxed threshold, so we allow more positions to enter into the model, so more putative QTLs to enter the model. And then we proceed a model optimization where we lower a little bit the threshold, so we are more stringent at this time, and we reevaluate every QTL, now conditional to the other QTL that were found in the previous step. So we keep doing this iteratively until no QTL is added nor dropped from the model. So once the model is stabilized, we have our final set of putative QTL. So that's our basic strategy. And just recapping the genetic model for a single family, we have the phenotypic observation. It is equal to the sum between to the sum of the intercept of the population, which is the population mean, the sum of all the genotype effects that belongs to all the QTLs in the final model plus the residual effects. As I mentioned, we use this genomic relationship matrix for all the QTLs, and we have one variance component associated with every different QTL. for the multi-parental population, we have a very similar model, but instead of having only one intercept, we have one intercept for each one of the subfamilies. And also we can have different variance components for all the QTLs within each one of the populations. And we can also make use of different variance covariance structures for the residuals. In our example, we have the three populations were evaluated in different, in very different environments. So it makes sense for us to use, to allow for different variance, residual variance components. So our basic strategy for those interconnected families would be performing the QTL mapping within families first. Then we perform the QTL mapping in the whole population. And then we evaluate the QTL for consistency across families. So just showing the results on the three populations here. The first trait with beta carotene, just let me explain this graphic first. So we have those bars here. Can you see the pointer there? Okay, so you have those bars here. Each one of those bars represent one chromosome. So we have those 15 chromosomes here. And we have different line colors here, which represent the population separate that were evaluated separately. So we have BT on red, TB on green, and NKB on blue. And the black line represents the joint analysis, so the results of the joint analysis. Those triangles here represent the QTLs that were identified in each one of those analyses. So as you can see, for the BT population, we identified two QTLs, one on chromosome number 3 and the second one on chromosome number 12. For the TB, the same QTL were identified at the same positions. And for the NKB, only the QTL on chromosome number 12 was identified. But for the joint analysis, both QTLs were identified. For dry matter, we have kind of the same, a very similar result where we identified two QTLs on the same chromosomes for BT. But instead, on NKB, we couldn't find any QTL for this trait. And for the TB population, we couldn't find the QTL on chromosome number 12, but we found signals on chromosome number 2, 9, and 15. And for the joint analysis, only the two major ones on chromosome number 3 and number 12. For the starch content, we could find the same QTLs on chromosome number 3 and 12 for BT population. Only the first QTL on chromosome number 3 for the TB, no QTL at all for NKB, and the two major QTL on the joint analysis. So that shows that the joint analysis has a higher statistical power to detect QTL. For this trait, we know that this trait is controlled by these two major genes, these two major QTLs or genomic regions, because we have evidence from previous studies. And we are now investigating how this kind of strategy performs in a more complex trait. So just showing that we can also account for the support interval, the QTL support interval, by using those results. So we take the peak and we calculate based on the p-value associated with those QTL peaks, where those QTL may be, what is the support interval according to a statistical confidence interval that we wanna use. Here we use 95%, and as you can see, all the QTLs for the three traits, they are overlapped, so they are probably the same QTL that control both traits. So all the three traits actually. So here you can see that as you imagine, so we have for these three traits, we have the same QTL controlling them. So there's a high genetic correlation between those traits. So between beta carotene and dry matter, we have a negative correlation of minus 0.57. Between beta carotene and starch, the same. And between dry matter and starch, we have a high positive correlation of 0.83. So that shows us that the results are pretty much consistent because we found the same QTL for all of them in the joint, essentially in the joint analysis, and supported by the individual population analysis. And also we got the estimates of the the variance components associated with the QTL for all positions and all the traits that were evaluated, and also the residual variances. So by making use of them, we can calculate the heritabilities associated with all the QTLs for all populations and all traits that were evaluated. As you imagine, BT was the only population that was able to detect those peaks by itself, not using the information from the other populations. So this is the population that has the highest values, the highest estimates of heritabilities, which range from 0.14 to 0.36 for beta-carotene. And the NKB population was the one that presented the lowest ones. For the dry matter, for example, we have 0 heritability, which makes sense because we didn't detect NQTL on this population by itself. But another thing we can do with this kind of analysis, with a joint analysis, is that we can calculate the allele effects for all the traits. And here we can check for consistency between those allele effects. So here I will show you the allele effects for beta-carotene. We have on the first, on this figure, we have the first graphic with the allele effects for the first QTL, chromosome number 3, and the second one for the allele effects on the QTL on chromosome number 12. So here the colors, the different colors, they represent the different founders. In this case, we have Beauregard as in red, New Kawogo or NK on blue, and Tanzania on green. The different shades, they represent the different homologs or alleles within this parent. So here, the 2 first colors, they represent the alleles, the estimates, the allele effects for the first population, the BT population. Here, the TB population, and here, the NKB. So as you can see, BT and TB, they have high magnitude allele effects, while NKB has lower magnitude allele effects, which makes sense because we have a lower value of heritabilities, and we couldn't detect this QTL in this population by itself. We could only detect it by making use of the joint analysis. And also the allele effects are pretty much consistent. As you can see, the fourth homolog on Beauregard, it has the positive allele effect for beta-carotene, for example, which makes sense because this is the parent that has the highest values of beta-carotene. And for the other haplotypes, the direction, both the direction and the magnitude makes sense for all the parents, and also for the Tanzania one. So we can make use of this to evaluate the consistency of the allele effects, and also to check even on NKB where we couldn't find or detect the QTL on the population by itself. We can see that there is a consistency of these effects when you can compare the direction of the allele effects with the other ones, with the other populations. And the same applies to the second QTL. And when you compare to the allele effects for the other traits, we can see the same. I just put it here. And I want to draw your attention to another thing, which is the negative correlation between beta-carotene and the other 2 traits. So if you take a look at the fourth haplotype on Beauregard, for example, you see that it has the highest magnitude in the positive direction, while for the other traits, this allele, this haplotype has a different signal, a different direction on the effect, which makes sense because they are negative correlated. So I just wanted to show you these results and draw your attention to the importance of being able to share the information between different families or different populations and make use of it to increase the statistical power and perform analysis where we can detect more QTL or more regions, and we can make use of these haplotypes for selection on populations, not even on biparent populations, but now we move our type of population analysis to a population that looks more with a breeding population instead of a biparent one. So that was what I wanted to show you. And Marcelo,
yeah,
like
yeah. So there's this will be like the next steps. So we were expecting to have data for a very interesting diallele an 8x8 hexaploid diallele. But we had some technical issues with the GBS part, and COVID happened, but hopefully we are going to get those, and we are going to try to expand this for more restrictive situations, because this, of course, this is very good data, and I think this is one of the best data that I work in all my life, because it's it's very complex, but it's still very clean, and in terms of development, the next step will be to combine different ploid levels. Actually, if you look into that function that is in my in this other branch in my GitHub, in the MAPpoly GitHub, you will see that this is already implemented. Everything here is already implemented, but we need to make it more user-friendly. Because if you have the whole hidden Markov model, it's just a matter of how you're going to visit your states. It's a matter of how you're going to show your states, because they are hidden, right? So that's what we did in this new version. So you can have, instead of just two parents with the same ploid level combined, you can combine whatever. And of course, for multi-allelic markers, the same thing, right? And we are also trying to make this more available for more complex population structures. We were working now with GKIN to make this work for rows. We have a huge diallele. It's a diploid, but it's a very wonderful diallele for acacia tree. But still, it shares the same problems that we have in polyploids, outcrossing high heterozygous individuals. And one of the things that we want to do is to collaborate with people that can help us with this, and because it's a very, very complex subject. And for Gabriel here, what you're going to do with QTLpoly?
Yeah, we will continue to develop QTLpoly and implement faster faster algorithm, so we can improve the computational efficiency. We will be implementing both the multi-parental analysis pipeline, and also we are planning to expand this for multi-trait and multi-environment QTL analysis. And as I mentioned before, we will be adding support for single-step models, so you can make use of this if you prefer to do that. Yeah. So yeah, that's our first.
Yeah.
We see.
And I want to, and this last slide is just an advertisement. So we have a position open at NC State, and I want to share with you, if you want to apply, this is a very interesting position where you can collaborate with a lot of the programs we have there. It's more oriented to the analytical part, but there will be a lot of practical things there. So, and we are open. And if you want more information, talk to me, or you can find the complete description of the position in this link here. Okay, so yeah, I think that's all we had to share with you guys. And thank you for your attention.